Welcome back to EC4 Educational Blog. I'm still remain on Kopi and I'm your tutor for biology. We're going to be looking at genetics part 2. If you're new in this channel, please don't forget to hit on the subscription button and share likes, and for comments you go to the comment section and ask your question, and then you will see SSCE, past question and answer, NECO, post UTME, and WAEC, and so on and so forth. Okay, today we're going to be looking at the second aspect of genetics, part 2. Here we're going to be looking at the principles of genetics, and then that principles of genetics we're going to be looking at these scientists that carry out this useful work on the field of genetics, which we call Gregor Mendel. This man is referred to as the father of genetics due to the useful work he did in the field of genetics. This man put forward 3 principles that explains the mechanism of heredity or mechanism of genetics. He put forward these 3 laws as follows. The first principle, law, is what he described as law of segregation or disjunction. And the second principle, law of independent assortments. And then the last is law of dominance. Okay, we're going to look at this law, and again in this video we're going to look at these principles and also look at some inheritance that deviated from this principle. There are some inheritance that did not obey this principle, so we're going to look at this principle that differ from this inheritance that differ from this principle, and also we'll look at this principle one after the other. I'm going to start with the law of segregation, a popular principle proposed by Gregor Mendel. Law of segregation, that is Mendelian's first law. Alright, so we look at this law. It states that genes or factors or alleles segregate or separates during meiosis, one from each parent prior to fertilization. This principle is very important. In fact, the principle is the most important aspect of genetics that cut across all other principles. These 3 principles are also under the control of the law of segregation. It is this law of segregation that tries to reconcile the number of chromosomes. Yes, the number of chromosomes in human is 46. 46. That is, these 46 are 2 pairs, which is 2n, that is when it is in diploid form. And then, if another organism also had 46, and then the number of chromosomes is still 2n, which is also in diploid form, that means how are we going to reconcile and keep a check on this chromosome to make sure that we maintain the same number of chromosomes? Is if this chromosome are halved, are segregated, are separated from each other, like this is going to be separated from n and n. So that means each number will now contain not 46 but 23 and 23. That is each number. And this thing happens during a process called meiosis. And the same thing happens here. We have n and n. So this is going to be a total of 4 daughter cells. So having 23 numbers and gametes. So when they now unite at random, the 46 number of chromosome is reconciled. As if this go with this, so it will be half of this still give me 46, or n and n still give me 2n. So to maintain this number of chromosomes, these genes must segregate during meiosis, and that is Mendelian's first law. But this Mendelian's first law most likely describes monohybrid inheritance. Yes, monohybrid inheritance is single factor inheritance. When Mendel carried out his useful work in genetics, he was an Australian monk. He did his study with garden. His experiment was with garden pear plants. And he had reason for choosing this garden pear plant. He believed that this plant can reproduce as fast as possible, and it can obtain what it can obtain in many generations in less than few days. In less than a few days to minutes to seconds, could obtain many generations at a go. That was the choice of garden pear plant. In this case, there are 3 traits he tried to study. One of the traits was the color of seed as well as the height. But let me use height as an example. I've done it in my first video. Mendel discovered that some plants, garden pear plants, that are predominantly tall and some were short. He tried to know what is responsible, why there are too many tall garden pear plants than short plants. He went to, on his own, pollinate these 2 different varieties. But at that time, he was studying only one single character. And he described that as monohybrid. So what he did was to get two parents. So I have a parental, like I discussed in my first video. When I talk about phenotype, I mean physical appearance of individual. So he was crossing a tall versus a short plants. And then from the genotype, which is the makeup of this individual, so the pair of tall allele is given as the sense dominant, and then it's given as the sense recessive. And then each gamete will only have one and not two pair. So what has happened so that one will be here and the other will be here. So this thing is called disjunction. That is that law. Disjunction or segregation. They have separated. That's what law means. And this must separate before they unite. So that means each gamete will have one cell, sex cell. And same thing happened here before fertilization cannot take place. And then that happened, we have this, the first offspring, we have this, the second offspring, we have this, the third offspring, and then we have this, the fourth offspring. It appeared that from the first video you will discover that any cross that is having a dominant and a recessive character, the dominant character will take precedence. So every offspring here are all tall, no shorts. To Mendel, this was so striking, but because he could not explain as of then what is responsible, why is it that this short gene did not appear here, and there were more tall plants than short plants. Out of curiosity, Mendel decided to self-pollinate. At that time, it can self-pollinate, it can self this one. So by selfing this, by getting this and this and mate them, when they have reached sexual maturity, get these two, self them because they are plants, so you can manipulate and self-pollinate this plant, and then it advances. It take it on to see if it can obtain several generations. This is the first. This is what we call first generation. So you can take these two and they becomes new parents. So if this is first parents, first parental phenotype and first parental genotype, then this could become parents here, and here he called them second parental phenotype. Remember that these two were now tall, so it is going to, what I say physically should be tall times tall. And then the second parental genotype was that they were this type of tall. They were this type of tall. So the law of segregation still remains sacrosanct. So this is going to go this way. We have this, and this, this, and this. And this is what the gamete is now. So each gamete will now have single. That's what it's called monohybrid. 1 versus 1. Mono gives this 1 versus 1. We get this 1 versus 1. We get this versus this. We get this. So this is what it called. If this is F1, so the F1 becomes parent. So the offspring of second parents becomes F2. This F2 means second filial generation. This means first filial generation. So in the second filial generation, Mendel discovered that this were tall, but not just tall, but they appear breeding tall. So I call it homozygote tall. So these two were more like heterozygotes because they are impure. They are not as pure as this, so it's still tall. Heterozygote tall. So this one is homozygotes shorts. So in terms of genotype, how many were homozygote tall is 1? How many were heterozygote tall is 2? How many was 1? So in terms of physical appearance, 1, 2, 3 were tall. So phenotypically, physically, 3 were tall, 1 is short. In genetics, this is what we call monohybrid phenotypical ratio. Anytime we cross a monohybrid inheritance, the ratio of the offspring, if they were heterozygotes pair and the mates, it's 3:1. And a summary of this first law is that Mendel discovered that in this principle of segregation, that law of dominance is sacrosanct. Law of dominance must be effective. What is the law of dominance all about? Law of dominance is that if you have a parent that is dominant against a cross of another recessive character, that the dominant character will always predominate in the first filial. That is what happened in the first filial between tall and short. Every offspring in the first filial were all tall. Short will not appear. This is where law of dominance this is where love is fully expressed. Then in the second, the short character will tend to appear in another generation because genetics is a transmission of character from parent to offspring from generation to generation. There are some traits that may not have passed on to the immediate generation, but they may tend to be seen in the other subsequent generations. So that is the case of recessive character. They may not be seen immediately in the third generation, but it's not as if they have died or they have extinct or they have gone into so many beliefs. But the fact still remains that since it is a character that is still transmittable, the fact that it is recessive, that does not mean that the gene is endangered or has extinct, but can come out after so many generations. It may be out in the next or subsequent generation. That is what happened in Mendelian's first law of inheritance as far as law of segregation is concerned. And then to the second principle, which is law of independent assortment.